And thanks to the speakers in the morning session to sort of lay the groundwork for the kind of biology that is possible with all the structures and all the method development that is sort of being catalyzed by having access to now millions and billions of structures, essentially at our fingertips. So in the afternoon, Gio, who is one of the main developers of iCn3D, he's going to tell us about the program itself. And this evening we have a workshop on iCn3D. So if you want to learn about this program, you can join the workshop around 6 o'clock. So people, I understand it sort of clashes with dinner time. So the people who join the workshop, you're also invited to the dinner right after the workshop. So anyway, so we wrote this grant with Philippe and Gio about creating an ecosystem around iCn3D, which is essentially a program that is web-based that allows us to not only visualize structures, but also analyze structures and share analysis just using a web link. So I actually joined this team as a user in about 2016. So this is what my lab does, essentially. So I'm a computational biochemist. We look at GPCRs. We're beginning to look at immunoglobulins. We also have some drug discovery and drug delivery projects. So I'm approaching sort of this type of ecosystem or these programs from a user's point of view and how we can contribute to this. And we are also developing methods to add to iCn3D. So in terms of the promise of personalized medicine, it's still not fully fulfilled, as you know. The challenge obviously has been that when you have mutations, it's very hard to predict the effect of these mutations given all the signaling networks and all the pathways that the proteins are involved in. So obviously the rigorous answer is if you understand at the structural level what's going on and the effect of mutations, then you can certainly predict the outcomes in terms of physiology and pathology, and hence develop treatments. So that's where programs like iCn3D and other computational methods come in that allow us to analyze structures. And when we look at the millions of structures available now, we need tools that can do a comparative analysis in a fast amount of time and sort of fast time that can then integrate structure information based on sequence and mutations and connect them to physiology, because a lot of the clinical data is available, but we don't have a direct link through structure and signaling to these linking sequence and physiology. Now, obviously, there are a lot of programs available to visualize protein structures. So our lab itself uses PyMol, VMD, and Maestro, but in terms of web-based programs, there are very few out there. iCn3D is one of them, and Molstar. So we'll learn more about iCn3D this afternoon. But this is what we are trying to do around iCn3D is to create sort of an open source ecosystem that will allow us to do both open science and using open data, and obviously creating this open source platform and expanding its reach. So this is sort of a quick overview of this platform, which is a web-based analysis and visualization platform. It brings in very interesting annotations, which are domain annotations from the CDD and other resources. So you can actually begin to start creating hypotheses around what different domains might be doing in the structural context. So Gia is going to go more into the detail of that, but I'm just going to provide a very brief overview of what the main features are. So as I already mentioned, it's web-based. It integrates sequence, structure, and now we are beginning to add functional information. It also performs automatic annotation, so you can bring in annotations from different sources. You can process large data sets, and that's what's going to be increasing over time, especially when we have all these millions of structures available very quickly. And one of the neat applications of this is obviously you can embed these structures and their visualizations in web pages, notebooks, as well as in publications. So we have already written recent publications where we provide structural visualization as a web link where you can just click on it and see the exact 3D view and rotate it around, like what we're discussing in that paper or publication. And then, obviously, as I mentioned, these visualization and analysis, the entire analysis can be shared by a web link. Now, so the vision that we have around this open source ecosystem that we're trying to create is obviously linking both genomics as well as proteomics through structure, and creating shareable molecular analysis and molecular scenes. What's going to be really remarkable is it will allow for a collaborative research mechanism between sort of collaborators, but also between instructors and students, because now students can interact with these structures like in a classroom setting, interactively, as well as through their even assignments that can be done interactively. And then what we want to create is an open source ecosystem around not only users, but also developers. So we are looking for users, developers, educators, collaborators who can help us create this ecosystem. And ultimately, we want to move towards a sustainable funding model. So NSF is catalyzing this, but eventually we need a more sustainable model of managing these open source ecosystems, as the audience in this sort of room would be fully aware that open source ecosystems might be easy to create, but they're harder to sustain over time. So this is the phase one that we have for our project, where we are connecting with users, both researchers and educators, as well as developers. We're seeking out new users. One of the things that we proposed was to create this scientific session. So that's why there's a check mark for that. So we will be organizing workshops and hackathons. We'll learn more about hackathons later on from Alyssa this afternoon. And we're beginning to engage with the PDB, as well as the EBI and UniProt and NCBI, obviously. And many of you would actually get maybe emails from us regarding interviews, so that we want to learn about what are the needs in the community around this area of visualizing protein structure and analyzing them sort of in an interactive fashion. We want to also look at experts in sort of software engineering, so that we can create a software development plan, because I'm a biochemist. I'm not a trained computer scientist, so we need good engineers that can create these platforms for like they're sort of future ready. And then we want to obviously engage with research labs as well and other open source organizations. And we are already beginning to engage with BOSC and 3D-SIG and other communities that are available around around ISMB. So this is a figure that NSF provided, because what they are envisioning is an ecosystem around an open source program like iCn3D. So they are catalyzing this for many different open source software in different areas. So for us around iCn3D, we need to create sort of a community that will have all these different components. So currently we have a team of collaborators, and it's growing. So you can see some of the names here. And we have collaborators like Barry and Bjorn Peters, and we have educators like Henry Jakubowski, as well as, and certainly we encourage like you to add your name here, so you can reach out to any one of us after the meeting, and we would love to talk about this. So in terms of acknowledgements, obviously NSF is funding this, and NIH has also contributed to supporting the people who are developing these programs.
Thank you very much.
Yeah.
I'm not sure we have one more question.
But we can take one or two. Yeah.
But we can take one.
Yeah.
As usual. Okay, so we will
so John is.
Sorry, John, go ahead.
You sort of listed it, but your goals seem a bit soft.
Okay.
Are there sort of real hard metrics you're proposing to achieve in terms of users, or I don't know what, new applications? How do you sort of define?
So the whole process of phase one is to define those concrete metrics.
Okay.
Yes, absolutely. Good question.